Welcome to the Global Addiction Therapeutics Innovation Day.
The next session, which will be focusing on molecular targets and biomarkers. And it gives me great pleasure to introduce Dr. Mehdi Farokhnia from NIDA in the United States. He also holds an adjunct position at Johns Hopkins, and he does preclinical molecular research and into substance use behaviors as models for substance use disorders, and he'll be speaking on some of that work today.
Okay, yeah, as mentioned, I'm at the NIH Intramural Program. And basically today I'm going to talk about some of the work we have been doing to integrate some rodent models with electronic health records from patients to enhance drug discovery for addiction. I'm mostly going to talk about alcohol use disorder, but basically this is a model we have been using for other addictions as well. I'm sure you all know, this audience, that alcohol use disorder is a chronic and multifactorial medical disease. People with alcohol use disorder have impaired ability to control drinking despite negative consequences, and that changes their brain and makes them vulnerable to relapse. There are treatments for alcohol use disorder. Again, this audience knows all of this. Broadly speaking, psychosocial treatments and also pharmacological treatments. We have off-label medications and some FDA-approved medications for alcohol use disorder. So the FDA-approved medications are disulfiram, acamprosate, and naltrexone. I put the year in parentheses here when they were approved, and you can see that it's way less than ideal. So on average, every 20 years, we have a new medication for alcohol use disorder. The number is very, very limited if you compare to other chronic conditions. So we clearly need better and more pharmacotherapy for addiction and also for AUD. But the challenge here is, as you all know, drug discovery is pretty lengthy and expensive. And it's even worse in neuropsychiatry and addiction. So it takes anywhere between 10 to 20 years to bring a molecule from preclinical models all the way to the clinic. And the story is very common that a lot of targets die between basic research and clinical research, right? This has been coined as valley of death. And I heard this quote a few years ago in a conference, and I keep putting it up because it makes total sense that we cure AUD in mice and rats every week in nature, right? So every week there is another paper coming out that we cured AUD, but then when we get to clinic, the success is way less than ideal. So again, our goal is to try to basically facilitate that crosstalk between preclinical models and clinical research. And a spoiler alert, I'm going to talk about GLP-1 that everyone is talking about these days as an example of how to integrate preclinical models with clinical research. If anyone doesn't know GLP-1 or glucagon-like peptide 1, it's a third amino acid peptide. It's produced in the intestinal mucosa and pancreas, and its main goal is to regulate blood glucose. So it's basically our satiety hormone. Unfortunately, these days, everyone knows about GLP-1, so I don't need to go through the details. But in addition to this well-known function that makes us full and regulates blood glucose, GLP-1 is also a neuropeptide. So GLP-1 is produced in the brain, the receptor is also produced in the brain. And without going into details, there is a lot of work showing that GLP-1 is involved in reward processing, for example, hedonic food intake, and also stress regulation, so interactions with HPA axis and cortisol. And that's why people have started looking at GLP-1 in the context of addiction. These are some papers that have been published from anecdotal reports. So this is where the media really started paying attention to these drugs, that people who are taking these GLP-1 medications like Ozempic, Wegovy, Mounjaro, things like that, they reported reduction in alcohol use and other addictive behaviors, right? But I do want to mention that this has been supported by decades of preclinical research. So again, not going into details, if anyone is interested, we reviewed the literature on GLP-1 and addiction in this recent review that you can look up. But overall, the data on the preclinical side is very promising. So with GLP-1 receptor agonists, reduce alcohol intake, alcohol place preference in preclinical models. One challenge, as you see on the right here, or maybe you don't see, is most of the preclinical literature has been with exenatide. So exenatide is a short-acting GLP-1 analog, which you have to, if you want to translate to humans, you have to administer like 3 times a day. But then more recently, there has been a discovery of more longer-acting GLP-1s like semaglutide and liraglutide. These are longer half-lives. They have higher affinity for the GLP-1 receptor, and they are more effective. So speaking of preclinical models, a few years ago, we started testing semaglutide and liraglutide in this paradigm, which is called two-bottle choice paradigm. If anyone is not familiar with this, basically you give a choice of water and alcohol to rats, and you ask them to self-administer. And we saw that semaglutide and liraglutide reduced alcohol intake. And semaglutide seemed to be more effective. And semaglutide is the generic name for Ozempic and Wegovy. I'm sure you have heard about semaglutide. These days it's everywhere in the news. It was a science breakthrough in 2023. It has revolutionized basically the treatment of diabetes and obesity. The developers got the Lasker Award, so it has been everywhere, and one of the most effective medications for obesity. So we started testing semaglutide in preclinical models. So we use different preclinical models. One is called drinking in the dark, which I know sounds sketchy, but it's basically you get mice to drink at binge level drinkings, right? During their dark cycle of the day, they really drink to a level of binge drinking, and then you can test the effect of different medications. So we tested different doses of semaglutide, and as you see here, both for sweet alcohol and unsweet alcohol, semaglutide reduced alcohol intake with a nice dose response. And then another model that we use is basically alcohol dependence. Here we put rats in cages and we make them, by exposure to alcohol vapor, we make them physically dependent on alcohol, and then you can do self-administration with them. And again, in both non-dependent and dependent animals, semaglutide reduced alcohol intake in rats. So super promising. We also had some other data, some electrophysiology that we published in this paper. And we were happy to see around the same time another group use a different model. So they use the two-bottle choice paradigm, and again, semaglutide reduced alcohol intake. So very promising preclinical data. But as I said, we have cured AUD in rats and mice 1 million times. So what we wanted to do is to transfer that to humans. And what we have been doing recently is using electronic health records, so data that already exist out there. So people are on these GLP-1 medications for other indications, right? And we wanted to see whether people who get GLP-1s for other indications would reduce their alcohol intake. For this study, we used some data from the veteran aging cohort study. There are different criteria. I won't go into details to make sure that you are really resembling a clinical trial. So you match people on different covariates. For example, here we matched on a bunch of covariates, and we used AUDIT-C, which is a measure of alcohol use severity. So these are the 3 questions I'm sure most of you are familiar with. You ask people how often they drink, how much they drink, and how often they binge drink, and that gives you a really good measure of alcohol use. And the beauty of this VA data set is they use AUDIT-C kind of as a vital sign. So every time people come in, they do an AUDIT-C. And we match people on a bunch of covariates, as I mentioned. We had people who were on GLP-1 drugs and those who were not. And here you look at difference in difference. Basically, you have a baseline AUDIT-C and a follow-up AUDIT-C, and you look at the difference between those who were on GLP-1s versus those who were not. I'm happy to talk about the details if anyone is interested, the statistical details here. And one beauty of this work is you work with really large numbers. So here in each of our groups, for example, as you see, we have 15,000 people after inclusion, exclusion, everything. So looking at the results, on the left, you see the overall difference in difference of AUDIT-C. As you see, that was significant, meaning that people who were taking GLP-1s, there was a significant reduction in their AUDIT-C score compared to controls. And then if you break down by AUD diagnosis, you see what you want to see. People with alcohol use disorder have a better response, and those with higher severity at baseline also have a much better response. So all promising. So far I talked about GLP-1 receptor agonists, but we have another system, basically another way of targeting the GLP-1 system, and that's inhibition of DPP-4. Again, without going into details here, DPP-4 is the enzyme that deactivates GLP-1. So if you inhibit that enzyme, you boost your endogenous GLP-1 levels. And those have been also approved for diabetes. So in this study, we also looked at DPP-4s. And as you see, the numbers were even larger because they have been around for a much longer period of time. And we looked at those who got the DPP-4 inhibitor versus those who did not. And here we did not see any results. So basically DPP-4 inhibitors, there was no effect, flat effect, there was no effect on drinking. And I keep telling the lab that this is like the only time that I was happy to see negative results because that's what we were expecting. That said, we went back, so this is an example of reverse translation. So we saw that in humans, but again, the electronic health records could be biased with a bunch of variables. So we wanted to take that information that we got from humans and back translate that to animal models. So we tested DPP-4 inhibitors in animal models. We tested linagliptin and omaregliptin. Linagliptin is peripherally restricted, so it doesn't go to the brain, and omaregliptin is brain penetrant. Not going to go through the details. We tested on the same paradigms that I mentioned. So first on binge-like drinking, drink in the dark. Again, there was no effect. You see on the left, there is no effect of linagliptin on drinking. Seeing that, we wanted to see whether the drug is actually working, pharmacologically active. And on the right, you see it does reduce glucose levels, so it is pharmacological active, but it's not reducing drinking. Seeing these results, we thought maybe the drug is not going to the brain, and so we tested omaregliptin, which is brain penetrant. Again, no effect. So consistent with our clinical data. Again, we kept being happy by seeing these negative results. And again, tested in rats as well, so in alcohol dependence model in rats. And again, no effect. So testing across different paradigms, different models, consistently, we didn't see any effect with DPP-4 inhibitors. So the summary of everything that I mentioned basically here is we concluded across mice, rats, and humans, GLP-1 receptor agonists are reducing alcohol intake, but DPP-4 inhibitors are not. And right now we are doing a clinical trial with semaglutide. We call it the STAR, semaglutide therapy for alcohol reduction. So if you don't know, semaglutide is a weekly injection. We are doing 2 clinical trials across the NIH Intramural Program on Oklahoma State University. It's a 20-week trial, so every 4 weeks we increase the dose. It's a weekly injection. We also do take control, which is a minimal behavioral treatment. And basically our primary outcome here is to see whether semaglutide will reduce alcohol intake. So self-reported drinking is our primary outcome. But since we bring people to the lab, we wanted to also do some experimental procedures. So 3 times during the study, we do alcohol cue reactivity. So we have this room at the NIH, which is built like a bar. It's a fake bar. I am a government employee, I have to say it. We don't drink there. We don't use taxpayers' money for drinking. But basically, we bring in people, we expose them to alcohol cues, we ask them to smell alcohol, and we ask them how much they are craving alcohol. And that has been a paradigm used in the field to see whether the medication would reduce people's cue reactivity as well. And another paradigm that we do is we have a virtual reality buffet. Basically, people put this headset, they go into this virtual buffet, and we ask them to pick the food that they want, right? So we wanted to see whether semaglutide also changes people's food choices. And last but not the least, while people are on the drug or placebo, we also do brain fMRI to see whether the drug is changing any activity or connectivity in the brain. We do different tasks like resting state, cue reactivity, monitoring incentive delay task, and other tasks to see whether the medication would change brain activity as well. I just put these up here to show that we are not the only group testing this drug. There are a number of other clinical trials. The first few are in academia in the US, the other one is in Denmark. And last but not the least, we were very happy to see a few months ago Novo Nordisk, the major pharma company that make Ozempic, they did start a clinical trial in people with alcohol use disorder and alcohol-associated liver disease. This is a gigantic multi-site clinical trial. Last time I checked, they have 200 sites around the world, and it was really happy to see a pharma company doing clinical trials in addiction. All that said, I put these up here. There have been a lot of anecdotal reports in the literature, and also a lot of attention from the media that people taking these drugs for other indications, reducing their alcohol intake or other addictive behaviors. And we get this question a lot. Should we start prescribing these for addiction? Should we advise people to take these medications for addiction? And as excited as we are, the answer is no from our perspective. We wrote this commentary a few months ago to just tell the community that yes, it's very exciting. We think it's one of the most exciting times in addiction medicine. We think we have a really good drug that could go up for approval, but we still don't have evidence from clinical trials. So we already have FDA-approved medications, and we have to stay for the evidence from these clinical trials to come out. And that's it. These are all the people in the lab. I want to thank. These are the people who do the real work. Yeah.
Wonderful, thank you. We have time for one question.
Sure.
Okay, can I? Yes.
Oh, sorry, I didn't see you.
Yeah, first, thank you. It's a very interesting information. And first of all, to say that besides Ozempic and Wegovy, there is also Rybelsus form, which is a pills form of semaglutide. I don't know whether you consider whether pills are involved as a form in this research, but we all well aware about the prices of this medication, and it's again about the accessibility for like majority of alcohol dependent people who can potentially need this treatment. But my question is, we also know about side effects, sometimes quite strong side effects. To what extent these risks are included in how you mitigate or how you manage this risk, and also risks of, because initially these medications are for gain weight or for diabetes, and are there any like risks in this sense if you use it only for alcohol dependence, but then person can have issues of other side? Yes.
Yeah, excellent question. Well, like any other medication, as you mentioned, they do have side effects, and I think we should be both okay and not okay with that, right? Like a lot of times people ask, I don't want to go on a medication and I don't want to shift my addiction from one drug to another drug. So, and we know that is not the case, right? But in terms of side effects, the most common side effects of these medications are non-specific gastrointestinal symptoms like nausea, if it's too much, some vomiting. So those are mostly manageable. In our hands, there are a minority of patients that they don't tolerate it, right? Like they start the medication, they take 1 or 2 injections and it's not for them and they stop. People from the obesity field say the same. But for the rest, most people have some nausea, some like change in bowel habits, things like that, which gets better and better actually by time. So people develop kind of tolerance to the side effects. There are the most common, the most serious side effect that we are concerned about, especially in people with alcohol use disorder, is risk of pancreatitis. That these drugs increase the risk of pancreatitis. People with alcohol use disorder are already at increased risk of pancreatitis. And that's the most serious side effect that we think can happen, but again, needs close monitoring. And so far, knock on the wood, like we haven't seen a pancreatitis in our trial because we are monitoring these people closely, and as soon as we see an increase in their amylase or lipase, we withdraw them from the study. In terms of weight loss, that's a very important point, that some earlier studies showed that these drugs are more effective on alcohol intake in people who were overweight as well, right? Some more recent data is like questioning that, but usually you don't want to start in people who are underweight for sure, because there's no buffer to go there. And then in the alcohol field, at least in the US, a lot of people with alcohol use disorder are also overweight. So we keep saying that this could be like killing two birds with one stone, hopefully. We are starting a clinical trial in collaboration with University of Maryland in people with cocaine use disorder and comorbid HIV. It's going to be a very, very small proof of concept study. But in that study, we are only enrolling people with a BMI of 25 or higher, because we know that people with stimulant use disorders, a lot of them are underweight, so we don't want to start with that. But these are all like open questions still in the field.
Organized by International Society of Addiction Medicine, sponsors and co-organizers, CNRE, Indivir, Spark Biomedical, Salofa, Ethifarm, University of Minnesota, European Union Drugs Agency, UK Office for Life Sciences,